Albert stain. Albert stain is used to demonstrate the metachromatic granules of Corynebacterium diphtheria. You will find that there are diphtheria is very common infection in pediatrics. Now let's see procedure. Fixation. The smear is head fixed. Smear is covered with Albert 1 stain for 5 minutes, then the excess is drained out. Albert 2 iodine solution is added for 1 minute. Slide is washed with water, blotted dry and examined under oil immersion field. Composition of Albert stain. Albert 1 comprises of toluidine blue, melachite green, glacial acetic acid and 95% ethanol and distilled water. Albert 2 contains iodine in potassium iodide. Interpretation. Corynebacterium diphtheria appears as green colored bacilli arranged in Chinese letter or cuneiform patterns with bluish black metachromatic granules at polar ends. So they appears green with their polar ends appears bluish black. This can be differentiated from diphtheroids which do not show granules and are arranged in palisade patterns. However, certain bacteria such as Corynebacterium xerosis, Gardnerella vaginalis also possess metachromatic granules. Other microscopic techniques. Other microscopic techniques include dark background and phase contrast microscopy for demonstration of spirochetes in genital specimens, hanging drop preparation for stool specimen for demonstration of darting mortality, giving a clue about V. cholerae. Acid fast organism and structures and percentage table. Now let's see other methods of direct detection, antigen detection, various immunological methods such as latex agglutination test, immunochromatographic test are available which detect antigen in clinical specimens. The classical example includes detection of capsular antigen of pneumococci, meningococci, H. influenza in CSF specimen, urinary antigen detection for pneumococci and Legionella, direct fluorescent antibody detection for detection of T. pallidum from most sections or exudates. Molecular diagnosis. Bacterial DNA or RNA can be directly detected by clinical specimens of various molecular methods such as polymerase chain reaction, culture identification and AST. Culture is the most common diagnostic method used for detection of bacterial infection. Specimens are inoculated onto various culture media and incubated. The colonies grown are subjected to identification and antimicrobial susceptibility tests. Now let's see culture media. A microbiological culture media is a liquid or a solid substance that contains nutrients to support the growth and survival of microorganisms. It consists of constituents of culture media are water and electrolyte, for example sodium chloride, peptone, It is a complex mixture of partially digested proteins obtained from various sources such as heart muscle, casein or fibrin or soya, agar. It is used for solidifying the culture media, does not add nutrient value to the medium. Sources. It is so constituent of culture media are water and electrolyte, peptones, which is complex mixture of partially digested protein obtained from various sources such as heart muscle, casein and fibrin or soya, agar, which is used for solidifying the culture media, does not add nutritive value to the medium. Source is it is prepared from cell wall of seaweeds and available commercially in powder form. Preparation. Agar powder is dissolved in water and temperature of the molten agar comes down. When the temperature of molten agar comes down to 45 degrees Celsius, it is poured into the petri dish and allowed to set for 20 minutes. Concentration. It is used in concentration of 1 to 2% for solid medium, 0.5% for semi-solid and 6% to inhibit protease swarming. Meat extract. It is commercial preparation of highly concentrated meat stock, usually made from beef. Yeast extract prepared from baker's yeast and malt extract which contain maltose. Blood and serum. They are important component of enriched media, provide extra nutrition to fastidious bacteria. Usually 5 to 10% of sheep blood is used. Alternatively, horse, ox or human blood can also be used. Type of culture media. Bacteriological culture media can be classified in two ways based on consistency. Culture media are grouped into solid media, semi-solid media and liquid or broth media. Based on the method of growth detection, culture media can be classified as conventional culture media. They are prepared from nutrients such as aqueous extract of meat, peptone, etc. Bacterial growth of is detected manually by visual inspection of turbidity or colony morphology. They are of various types based on their functional use or application. Simple or basal media, enriched media, enrichment broth, selective media, differential media, transport media, and anaerobic media. Automated culture media. They are mainly available for blood and sterile body fluids. The growth is detected automatically by the equipment. Conventional culture media. Simple or basal media. They contain minimum ingredients that support the growth of non-fastidious bacteria. Example includes peptone water. It contains peptone which is 1% and NaCl 0.5% plus water. Nutrient broth. It is made up of peptone water plus meat extract 1% and is available in three forms: meat extract, meat infusion and meat digest broth. Nutrient agar. It is made up of nutrient broth plus 2% of agar and semi-solid medium. It is prepared by reducing the concentration of agar 2.2-2.5%. A is the figure of peptone water, B of nutrient agar, C of blood agar and D of chocolate agar. Uses of basal media. The basal media are used for testing of non-fastidious bacteria. They serve as a base for preparation of many other media. Nutrient bath is used for studying bacterial growth curve. Nutrient agar is preferred media for performing the biochemical test such as oxidase, catalase, slide agglutination test, etc. to study colony morphology, pigment demonstration. Semi-solid media is used for demonstrating motility of the bacteria. Motile bacteria spread throughout the semi-solid media, making of medium hazy, maintaining the stock culture. Enriched media. When the basal media is added with additional nutrients like blood, serum, or egg, it is called as enriched media. In addition to non-fastidious organisms, they also support the growth of fastidious nutritionally exacting bacteria. Example includes blood agar. It is prepared by adding 5 to 10% of sheep blood to the molten nutrient agar at 45 degrees Celsius. It is most widely used media in diagnostic bacteriology. Blood agar also tests the hemolytic property of the bacteria, which may either be first partial or alpha green hemolysis, complete or beta hemolysis, described subsequently in the chapter. Chocolate agar. It is heated blood agar prepared by adding 5 to 10% of sheep blood to the molten nutrient agar at 70 degrees Celsius so that RBCs can be lysed and the content of RBC will be released, changing the color of the medium to brown. It is more nutritious than the blood agar and even supports certain highly fastidious bacteria such as Haemophilus influenzae that does not grow in blood agar. Loeffler's serum slope. It contains serum. It is used for isolation of Corynebacterium diphtheria. Blood culture media. They are also enriched media used for isolating microorganisms from the blood. They are available either as conventional or automated blood culture media. Enrichment broth. They are liquid media added with some inhibitory agents which selectively allow certain organisms to grow and inhibit others. This is important for isolation of the pathogen from clinical specimen which also contain normal flora. Example includes stool and sputum specimens. Example of enrichment broth includes tetrathionate broth used for Salmonella typhi, gram-negative broth for isolation of Shigella, selenite F broth for isolation of Shigella, alkaline peptone water used for Vibrio cholerae. So enrichment broth is a liquid media with some inhibitory agent for selective growth of organism and inhibition of others. This is important for isolation of pathogen from clinical specimens. It also contains normal flora. Selective media. They are the solid media which contains inhibitory substances that inhibit the normal flora present in the specimen and allow the pathogen to grow. They inhibit the normal flora present in the specimen and allow the pathogen to grow. For example, Lowenstein junction media, which is used for isolation of Mycobacterium tuberculosis, thiosulfate citrate bile salt sucrose, TCBS agar, which is used for isolation of Vibrio species, DCA, deoxycholate citrate agar, and XLD, xylose lysine deoxycholate agar, which is used for isolation of enteric pathogen, for example, Salmonella and Shigella for stool, potassium tellurate agar, used for isolation of Corynebacterium diphtheria. Lowenstein junction media, this is figure A is LJ media and figure B is TCBS agar. Transport media, they are used for transport of clinical specimens suspected to contain delicate organism or when delay is expected while transporting the specimens from the site of collection to the laboratory. Bacteria do not multiply in the transport media, they only remain viable. Differential media, this media differentiates between two groups of bacteria by using an indicator which changes the color of colonies of a particular group of bacteria but not of the other group. For example, McConkey agar. It is a differential and low selective media commonly used for isolation of enteric gram-negative bacteria. It differentiates organism into lactose fermenter produce pink colored colonies, for example Escherichia coli, and non-lactose fermenters which produces the colorless colonies. The composition of McConkey agar is peptone, lactose agar, neutral red indicator, and taurocolate. Most laboratories use combination of blood agar and McConkey agar for routine bacterial culture. So what happens when the lactose fermenter bacteria are grown in McConkey agar? They will impart pink color, and when they are non-lactose fermenter, the original yellow color will remain. So why the lactose fermenter will impart pink color? It is because of the formation of lactose as lactic acid, which gives red color on neutral red indicator. Now let's see clade agar. Cysteine lactose electrolyte deficit agar. Another differential media which is similar to McConkey agar and is capable of differentiating between lactose fermenter and non-lactose fermenter and is used as an alternative to combination of blood agar and McConkey agar for the processing of urine specimen. Figure A of DCA, Figure B of XLD, Figure C of McConkey agar and Figure D of clade. Let us see anaerobic culture media. Anaerobic culture media contain reducing substances which take up oxygen and create the lower redox potentials and thus permits the growth of obligate anaerobes such as Clostridium. Examples are as follows: Robertson scoop meat broth. It contains chopped meat particles, beef heart, which provide glutathione, a sulfhydryl group containing reducing substances, and an unsaturated fatty acid. It is most widely used anaerobic culture media. It is used for maintenance of stock cultures. So, Robertson cooked meat broth contain meat particles which provide glutathione and unsaturated fatty acids. Other anaerobic media are thioglycolate broth, BH1S agar, brain heart infusion agar with supplements like vitamin K and hemin, neomycin blood agar, egg yolk agar, phenylethyl agar, and bactericides bile esculin agar. Blood culture recovery of bacteria from blood is difficult as they are usually present in lesser quantity in the blood and many of the blood pathogens are fastidious. Therefore, enrichment media is used for isolating microorganisms from blood. Blood culture media are available either as conventional or automated media. Conventional blood culture media are of two types: monophasic media, which contains brain heart infusion broth, and biphasic media. It has a liquid phase containing BH1 broth and a solid agar slope made up of BH1 broth. Figure A represents Robertson cooked meat medium. Figure B thioglycolate broth. Figure C is brain heart infusion broth, which is monophasic media, and figure D is biphasic media, which contains both solid as well as liquid broth. The recovery of the organism in the blood is enhanced by mixing the blood in the broth periodically. If any growth occurs, it can be detected by subcultures. Disadvantages: in conventional blood culture, subcultures are made manually. This process can be performed less frequently, once a day, and it is cumbersome. From monophasic BH1 broth, subcultures are made onto the blood agar and McConkey agar periodically for 1 week. There are high risk of contamination due to opening of cap of the bottle every time when the subcultures are made. From biphasic BH1 broth, subcultures can be made just by tilting the bottles so that the broth runs over the agar slope. There is lower risk of contamination as it obviates the opening of cap in the bottle. Automated blood culture. Automated blood culture techniques have been in use since last 2 decades. They are revolutionary, offer several advantages over conventional blood cultures. Continuous automated monitoring. Following inoculation, the culture bottles are loaded inside the automated culture system. The incubated bottles are periodically tilted automatically every 10 minutes, which allow the mixing of blood with broth, which fastens the recovery. Bottles are periodically monitored for the microbial growth once in every 10 minutes by the instrument. Once positive for microbial growth, instrument gives signal producing beep or color change on the screen. Composition. Automated blood culture bottles contain tryptic soy broth and or brain heart infusion broth as enrichment media added with polymeric resin beads which absorb and neutralize the antimicrobial present in blood specimens. In addition to blood, the bottle also be used for culture of bone marrow, sterile body fluids such as CSF, peritoneal, pleural, and synovial fluid. More sensitive, it gives higher yield for positive culture from clinical specimen. Rapid. It takes less time than conventional method, less labor intensive as fully automated. Automated systems that there are 3 automated systems commercially available: Bactec or Alert 3D. So this is the Bactec or Alert automated blood culture system. Bacti or Alert 3D, its principle is based on colorimetric detection of growth. When bacteria multiply, they produce CO2 that increases the pH, which in turn changes the color of blue-green sensor present in the bottom of the bottle to yellow, that is detected by colorimetry. Bacti or Alert Virtuo Biomeriux. It is an advanced form of Bacti or Alert which offers several advantages such as automatic loading and unloading of bottles, faster detection of growth, can determine the volume of blood present in the bottle. So this is the figure of Bacti or Alert Virtuo automated blood culture system. Now let us see Bactec BD diagnostics. Its principle is based on fluorometric detection of growth, use of oxygen sensitive fluorescent dye present in the medium. In an uninoculated medium, the large amount of dissolved oxygen is present in broth quenches the fluorescent dye. Later, actively dividing microorganisms consume the oxygen, removing the quenching effect and allowing the fluorescence to be detected. There is an automated culture system available for culture of Mycobacterium tuberculosis from various pulmonary and extrapulmonary specimens called as Mycobacterium growth indicator tube MGIT. This works on fluorometric principle for detection similar to Bactec. Disadvantages: automated culture methods do have several disadvantages like high cost of instrument and culture bottles, inability to observe the colony morphology as liquid medium is used. Culture methods. Culture methods involves inoculating of specimen onto the appropriate culture medium, followed by incubating the culture plate in appropriate conditions. So let us again see the disadvantages because it is often asked. Automated culture methods do not have several disadvantages like high cost of instrument, do have several disadvantages like high cost of instrument and culture bottles, inability to observe the colony morphology as liquid medium is used. From culture method we will see in next video.